The organism and trait we are representing here are penguins and their feather color, which is controlled by eumelanin.
Transcription starts in the nucleus of a cell, and its purpose is to make mRNA form a strand of DNA. The polymer of RNA is nucleic acids and the monomer is nucleotides. Unlike DNA, RNA consists of phosphate group, ribose sugar instead of deoxyribose, and one of the 4 nitrogenous bases, guanine, cytosine, adenine, and uracil, as opposed to thymine in DNA. In the initiation phase, the DNA is inside the nucleus along with different mRNA nucleotides floating around. Then, an enzyme called RNA polymerase attaches to the promoter region and begins the translation process. Inside the DNA polymerase, the DNA is unzipped and the RNA nucleotides slide along the template strand to create the complementary strand. Then, when RNA polymerase reaches the terminated region, the mRNA strand and RNA polymerase detach and get ready for processing.
mRNA processing. In the completed mRNA strand, only some parts are necessary to move on to translation. The parts that are cut out are called introns, and the parts that stay are called exons. These parts are involved in a process called intron splicing. The intron are cut out, and the exons are put back together in one chain. Additionally, a 5' gap and a 3' poly A tail are added to each end. The mature mRNA strand then leaves the nucleus for translation.
Translation occurs in the cytoplasm of a cell, and its purpose is to make a protein from the mRNA sequence. The mRNA then attaches to a ribosome, which is composed of rRNA or ribosomal RNA and proteins. It slides into the ribosomes, and they then read it in 3 nucleotide sections called codons. When the ribosomes read the start codon of AUG, it will start bringing in amino acids connected to anticodons, which are the complement to the codons. The amino acids and the anticodons are on tRNA or transfer RNA. When the tRNA comes, another one comes after, and the 2 amino acids bind together with a peptide bond. Once the 2 amino acids are bonded, the first tRNA leaves and the rest slide up, and another tRNA takes the second one's place, and this process continues until a polypeptide chain is formed. Once the ribosome reaches the stop codon, in this case UAA, it stops bringing in amino acids and the structures dislodge from each other, leaving a polypeptide, which then forms into protein, in this case eumelanin.
Protein. The protein we demonstrated was eumelanin, which controls the color of the feathers of the penguins.
One type of mutation that can occur is substitution. For example, if the original DNA sequence reads TAC, CAT, ACT, and the complementary mRNA sequence reads AUG, GUA, UGA, then the amino acid sequence would be MET, VAL, and then it would stop. However, if the mutated DNA sequence reads TAC, CTT, ACT, and the complementary mRNA sequence reads AUG, GAA, UGA, then the amino acid sequence would be MET, GLU, stop. Therefore, the mutation is a missense substitution mutation because it changed the amino acid sequence, and consequently the protein would not be the same and the trait could be lost or changed.